thank you all so much for joining today for the first Wolfson Green Talk of the academic year 2022-2023. Today we have our speaker, who is Dr. Yang Liu, who is a research associate at the McDonald Institute for Archaeological Research, as well as a Wolfson College research associate. Yang is currently in Australia, having just moved out there. So thank you to Yang so much for joining us and making good use of your jet lag time. I believe Yang mentioned it's 3 a.m. out there. This talk is part of the Sustainability Conservation Hub and Science Society collaboration, part of the Cambridge Green Week happening this week. Yang, being an evolutionary geneticist, is broadly interested in eco-evolutionary dynamics of plant populations that have undergone environmental changes to their genes through long time and through location. Yang's research aims are to provide a holistic understanding of the major episodes in plant demography and life histories that can then be used to inform conservation interventions and foster more sustainable practices in connected fields. His work is at the interface of ecology, evolutionary biology, and the interplay of genetics and genomics, so not contentious at all. Thank you so much, Yang, for joining us today. And with that, Yang, how to forecast organism adaptability to future climates? Over to you.
Thanks, Charlie, for that introduction. Good evening, everyone. Thanks for taking the time out of your busy day to listen to this talk. Today, I will be presenting on the prediction of climate adaptation, digging into some of the work that I have done over the past 7 years, all the while trying to assess the adaptability of forest trees and contemporary climate change using genomic information. Prior to all, I want to offer you a conceptual framework about my research motivation. Biodiversity is disappearing at an unprecedented rate and climate change. A synthesis of 113 published studies give an estimate of about 8% of species becoming extinct from climate change. Moreover, this extinction rate would be accelerated with the global temperature rise. For example, a temperature increase by 2 degrees Celsius, 1 in 6 species could perish. Prior to the loss of species, their geographic distribution ranges must have shrunken. Of about 1,000 species surveyed, 15% species are subject to shrinking distribution ranges. This implication of global biodiversity loss over time is that genetic diversity within species is progressively being lost before species become extinct. That said, there must be many species that are becoming genetically depauperate, leading to the global gene pool decaying faster than the loss of species. Such a notion has motivated a means to assess putative genetic maladaptation by using genomic and climatic information. If the pattern of genetic diversity decay can be successfully projected by climatic predictors, then we can adopt countervailing conservation practices such as assisted migration or gene flow. Through such interventions, we eventually hope to prevent climate-related loss of biodiversity. I have structured the talk in the following way. I will first talk about a space for time approach implemented for adaptation research. Based on that approach, I will then turn to the prediction of genomic vulnerability of populations and the climatic scenarios. Finally, I will turn to the assessment of the adaptability and the evolvability of phenotypes in a changing climate. First of all, what is environment adaptability? It means evolved genetically based phenotypes that confers a fitness advantage in a particular context. The central thrust of this definition is twofold. One, it deals with phenotypic traits that have fitness relatedness. And second, these phenotypic traits have genetic basis and are able to evolve. The fitness environment relationship can be illustrated mathematically by a normal distribution shape. Given a population which can attain the highest fitness at the environment A, we say the population is adapted to the A environment. When the environment shifts backward or forward, the population is perturbed off the fitness peak. So the population is maladapted to any environment but the A. In general, there are three main fitness components: survival, reproduction, and growth. Fitness-related traits can be used to indicate aspects of fitness. Assuming that the solid curve describes the current realized trait distribution of an adapted population. If we want the population to remain in the environment optimal in the future, the trait distribution needs a shift, for example, to the dashed line curve. The shaded gray area indicates the risk of maladaptation. So in this case, if many individuals of the population express low trait values in the future, prompt its potential maladaptation. Ideas draw on the interactions among fitness, phenotypic traits, and climate prompt the use of mathematical approaches to characterize their relationships. For example, the use of the current climate information as a function of a phenotype to construct a predictive model and then project the phenotype under given climatic scenarios. Determine statistical relationships between species occurrence data and climatic predictors to track ecological niche suitability under changing climatic scenarios. These predictive models can be applied to extrapolate across climatic landscapes. Here is an example depicting a life history trait of a pine tree predicted across British Columbia at present. And in the 2015s, using selective climatic variables. As you can see, compared to the 1918s, the study trait is inclined to increase in southern British Columbia in the 2015s. And another example portrays the ecological niche suitability of popular trichocarpa in the Pacific Northwest. Based on the current scenario, we infer the species niche suitability in the past, last glacial maximum, and a future time period, for example, in the 2015s. So we can see there are some changes in the past and in the future compared to the current scenario. Nonetheless, such models assume statistical trait-climate relationship or environmental tolerance stasis. But phenotypic selection and population divergence have been occurring all the time. To overcome this hurdle, we naturally think of incorporating the evolutionary notion in the model. Let me introduce two experimental designs first. So first is reciprocal transplant experiment. So in this experiment, individuals are reared in both their home and reciprocally in a foreign environment. This experiment can be used to test for local adaptation by assessing differences in survival and reproductive success in the two environments. The second approach is common garden experiment or space for time approach. Individuals are reared in identical environment conditions. In doing so, we expect to disentangle the effects of genetics and the phenotypic plasticity and phenotypes. Through a space for time experiment, we assume that what we observe across space also holds true over time. So given data gleaned from a common garden test site, as shown on the map, we use genomic, then we plant the propagators in common garden test site. We use genomic scan to identify associative genetic markers or adaptive genetic markers. That is, the genetic markers significantly associated with phenotypes or environmental variables. Based on these selective markers, we can implement computational approaches to projecting their evolutionary consequences. Predicting population genomic vulnerability is emanated from this concept. The following section will present 3 approaches to genomic vulnerability prediction. First, for example, this is what we have done in a recent project of popular trichocarpa. We sampled 409 individuals from 29 drainages across the species distribution range, gray shaded on the map. Then propagators were grown in a common garden site. After 8 years of acclimation to the field plant site, we measured 27 phenotypic traits for 3 consecutive years. Correlation between temporal environment of the common garden and the traits related to growth and or biomass or phenology are at an intermediate level, around 0.4 to 0.45, indicating strong plastic phenotypic changes in time. Meanwhile, you can see that most of the traits averaged across years display a gradient variation with latitudes of origin. This suggests a strong genetic variation in plastic phenotypic changes. By performing genotype-phenotype environment association analysis for the previous 409 individuals, we identified a set of peak associated markers under selection by climate. Then we use spatial simulations to foresee population genetic structure and future climatic scenarios. Here is what we got. 8 colors represent 8 genetic clusters. The first panel is the current scenario. The others are the current scenarios encompass 3. So the others are just the forecasted population structure and the different greenhouse gas emission scenarios in the 2015s. The species of the current scenario encompass 3 main populations: North, Central, and South, green, blue, and red. However, climate change would substantially affect population genetic structure along the latitude distribution of the species, resulting in a possibility of homogenizing the central and south populations. This indicates that the south population will be likely undergoing the strongest selection and climate change, and may be the highest, maybe of the highest vulnerability. Compared to the preceding intraspecific turnover by genetic clusters, the genetic offset estimates allow for predicted genetic component turnover by location. So how does this work? Just anchor yourself in this figure. A shift along the climatic gradient, say from low to high temperatures, could be due to a temporal change in climatic conditions for a focal location. These climatic differences associated with a corresponding shift in the adaptive genetic component, represented by adaptive genetic markers from genotype environment association analysis. The magnitude of this shift is quantified by genetic offset. For example, a change from A to A primer on the climatic gradient occurs in a region of high turnover in the adaptive genetic component, yielding a large predicted genetic offset, GAA primer. Whereas the same change on climate between B to B primer produce a small amount of predicted genetic offset, GBB primer. Moving forward, we can visualize the results across space. A and B represent two locations under the current climatic scenarios, while A and A primer represent the climates in the same location in the 2050s. We can easily calculate genetic offsets between these two time periods for A and B locations. Likewise, perform such a calculation across the entire species distribution range. By doing so, we estimate genetic offset, genetic turnover across sites. The map reveals many range age sites in the north are likely subject to high genetic turnover in the future. So the previous two methods provide means to predict the genetic turnover under climate change. But what if we aim to help potentially maladapted populations get adapted to future climates? Assuming that we have identified a set of adaptive genetic markers under selection by climate. So x-axis is a climatic variable. Y-axis is population allele frequency of adaptive markers. Each dot represents allele frequency of a population. By regressing on an environment gradient, we get a genotype environment optimal. A focal population in blue has a current risk of non-adaptiveness due to its wide deviation from the optimal line. We have two possible adaptive strategies. First, assisted migration, a translocation of this population to a location with a more suitable climate. Second, assisted gene flow, move of genetic material from donor population to a location with a more suitable, with allele frequency better suited to the focal population environment. As climate change, the donor population alters, which depends on the magnitude of climate change at this location. So identifying an ideal donor population is comparable to shifting the recipient population horizontally until its future climatic position, and then finding corresponding populations on the genetic trait. Here is a proposed assisted migration or gene flow in one study of dwarf birch in the UK. The left base map is mean annual temperature. The right map is based on mean diurnal range. Using different environment predictor may generate different gene flow strategies, shown by arrows for movements, directions, and donor populations on the map. So you may be wondering whether the prediction of genomic vulnerability can reflect the true adaptability and evolvability of populations. I propose a direct assessment based on phenotypes. So the third section uses a case of logical pine for demonstration. Here is the use of 4 test sites for experimental simulation. According to mean annual temperature, we define Judy and Virgin are 2 warm test sites. Swan and Time are 2 cold test sites shown by the location icon in red and blue. Moreover, Verge and Time are more humid in each group. Similarly, we classify seed source populations into warm red and cold blue categories. After multiple years establishment for the common garden test sites until trees at maturity, we chose about 115 trees from the test sites and measured 4 focal traits, including height growth as a proxy for fitness, carbon isotope ratio, delta carbon 13, hydraulic conductivity measure as an indicator for drought avoidance, resistance to 2 pests. One is severity of West Gore rust based on quantitatively scoring West Gore rust symptom on trees. The other is suitability to mountain pine beetle evaluated by quantifying defense chemicals. When evaluating the adaptability of phenotypes, we use delta mean annual temperature as the environmental predictor. Delta mean annual temperature equals to mean annual temperature difference between a test site and a population origin site over a reference normal period. We posit 4 scenarios for the adaptability evaluation. Given a population trade mean across test sites, if trade values increase with increasing delta mean annual temperature, then high trade values can be expected for cold origin populations, and thus high adaptive capacity for cold populations. This is scenario 1, black field. Scenario 1, white circled. Given test site trade means across populations, as delta mean annual temperature widens, trade values decrease, indicating high trade values in cold test sites. Opposite to these 2 scenarios are scenarios 2, black and right. We hypothesized as such. We then we are showing that height and delta carbon 13 pertain to scenario 2, black and white, indicating warm populations and warm test sites produce a higher adaptability of both traits. It seems that West Gore rust matches scenario 1, black and white, whereas mountain beetle only matches scenario 2, black. This suggests that West Gore rust and mountain pine beetle have an opposing pattern of adaptability along the climatic gradient. To test evolvability, it is imperative to assess the strengths of selection. First, we consider 2 types of selection: linear or directional selection in a negative or positive manner. Quadratic selection, including stabilizing and divergent selection. Coefficients of the fitness trait relationship are selective strength, beta or gamma. Beta values generated from the linear selection, gamma values generated from the coefficients of quadratic selection. So as we use height as a surrogate for fitness, thus it is not possible to estimate its selective strength. So that's why you can only see the selection evaluation for 3 traits without height included. Let's look at the results. The first column of the heat map is beta, while the second column is for gamma. Red and blue for positive and negative values, respectively. We tested side by side, and all sides combined. We can see that the direction of selection doesn't change in all test sites of each trait, but the selection differs in magnitude between sites. Cold sites appear to select for drought avoidance with a great intensity. All pest traits are selected against or are negative values. Next, we estimate response to selection in each test site, which is the product of among trait genetic relationships and trait selective strengths. We adopted a multivariate approach. The networking connection showcases the relationship among the 4 focal traits in one test site, and the line width is in proportion to the selection strength or the correlations between traits. As no selection analysis performed for height, we assume 2 scenarios: lower selection in height relative to delta carbon 13, say the low scenario, or higher scenario in height relative to delta carbon 13, say the high scenario. We use an array of selection coefficients of height for each scenario for simulations. So for the no scenario, we can see that response to selection delta Z increases for height and drought avoidance in warm than in cold test sites. However, for high scenario, just to say for the red boxes and lines, delta Z of drought avoidance is lower in warm test sites than cold sites, whereas height remains more selected for in warm test sites. With regards to pest severity or suitability, the overall delta Z is much weaker than height or drought avoidance, and both pests are selected against negative values. The 2 cold test sites have relatively high delta Z for both West gore rust and mountain pipe beetle, whereas a warm and humid climate at virgin leads to the high delta Z for West gore rust and lower for mountain pipe beetle. Similarly, a warm, dry climate at Judy results in the lowest delta Z of West gore rust, but relatively high for mountain pipe beetle. This suggests that rainfall affects response to selection of West gore rust versus mountain pipe beetle under warming. So I conclude by outlining 3 key messages. First, I accentuate the use of space for time substitutions, mimicking future environment changes. Second, I strongly advocate for the implementation of genomic-based projections for population vulnerability by using multiple approaches to work out reliable intervention strategies for assisted adaptation. Third, I propose experimental approaches to assess adaptive capacity and response to selection of phenotypes to validate the preceding genomic predictions. Just to thank the many partners and funders that have made all of this work possible. With that, I'll hand it back to Charlie to moderate potential questions.